Hi, everyone. So welcome back from your break. And if you're just joining us, welcome. My name is Arnika Flandrin, and I'll be the moderator for this second group of talks on genetics and molecular biology. As a reminder, please enter questions for the speakers into the chat window. And also speakers, please know that you will hear two bell sounds during your presentation. One will be 5 minutes prior to the end of your talk, and the other will signal the end of your talk. So we can go straight into the presentations. So our first talk in this section is from Dr. Michael Telius. He's a postdoctoral scholar here at UC Berkeley in Dr. Rich Kramer's lab. Dr. Telius received his bachelor's and master's in medical sciences and medical neurobiology, respectively, from the Hebrew University in Jerusalem, Israel. He then received his PhD in cell and developmental biology from Tel Aviv University. In his talk today, he will present his research on the molecular mechanisms behind ganglion cell hyperactivity in degenerating retinas and the therapeutics that may be effective in treatment. Please welcome Dr. Telius.
Thank you very much. Thank you for having me. It's an honor. So I'm going to jump right to the talk. You know perfectly well what happens during degeneration of the retina in cases such as retinitis pigmentosa or age-related macular degeneration. Basically, the photoreceptors die. Can you hear me? Because I have a problem here. Okay, yeah. And the photoreceptors die in the outer retina, and of course, there are many efforts in trying to either implant new cells or slow down their death, but I am going to focus on what happens down here in the inner retina, and specifically in the RGCs, because the RGCs, as you know, form the optic nerve, and all the input in the retina has to go through them to reach the brain. And what we know, we have known for years, is that as we lose our vision because of degeneration in the outer retina, because of the death of rods and cones, we also get a pathophysiological remodeling of the inner retina, and specifically in the RGCs, whether it's halfway there or especially after total degeneration. These cells become hyperactive and hyperpermeable, as I will show you in a moment. And this is a problem because this hyperactivity obscures light responses, but also will prevent or will reduce the fidelity of the signal when we want to restore vision by using optogenetics or prosthetic devices, etc. We have looked at the issue of remodeling of the inner retina while the photoreceptors are dying, and we and others have described two major events that happen there. These are MEA recordings, multiple electrode array recordings of RD10 mice, and you can see that at P14, this mouse has perfectly good vision, all the on and off responses are there, and as the mouse ages, the photoreceptors die, the light responses dwindle, but also you can see that the baseline activity of the RGCs increases dramatically. You can see that with lowering light, worsening light responses, that spontaneous hyperactivity goes up. You can see here how in a wild-type animal, we don't see that, and in an RD1 animal, which has a mutation that is even more severe than the one in RD10, you see that pretty early in degeneration. We also show here that this spontaneous hyperactivity of the RGCs, which is 6-fold, 8-fold higher than the one in the wild type, is not dependent on synaptic input. When we block all the synaptic inputs to the RGCs and isolate them and record only from them, we still see this hyperactivity. The second phenomenon that we see happening early on in degeneration, when the photoreceptors out there are dying and the inner retina down here is sensing that death, is that these cells become hyperactive. Dyes, in this case is FITC, we can use also YOPRO dyes. These dyes that are excluded from the wild-type retina. The cells these dyes don't penetrate the cell membrane. They do so in the degenerated retina. You can see here the axons of RGCs. So this is also happening specifically in RGCs. And our question is a fundamental question. What is the signal that is coming, that is happening out there in the outer retina and is reaching all the way to the inner retina, to the RGCs, and changing them profoundly? And as I said before, this has a lot of consequences for vision restoration and for obscuring light responses during degeneration. And to make a very long story short, I will say that what we have found is that this is caused by RAR. RAR is the retinoic acid receptor, which is activated by retinoic acid. And we have demonstrated in a paper that we published in Neuron last year and another one this year in JCI, how when photoreceptors die, retinoic acid invades the inner retina, activates its receptor in the RGCs, maybe in other inner retinal neurons, but for sure in RGCs, increasing the spontaneous firing and reducing signal to noise ratio. But when we block retinoic acid, we reduce that background noise and we can improve vision. So the first task that we wanted to do was to show that indeed retinoic acid receptor activation is increased in degenerated retinas as opposed to wild-type retinas. So we developed a genetically encoded reporter. This is the retinoic acid receptor binding to its ligand, retinoic acid. This is a transcriptional activator, an enhancer of transcription that binds directly to DNA at specific sequences known as RAR, retinoic acid response elements. And when we hack that system basically and put that those sequences upstream to a GFP, what we have is a reporter for retinoic acid mediated transcription. We also, for good measure, put another fluorescent protein there, a red one, to indicate which cells have been transfected or not. You can see here the results in vitro, in HEX cells, how only upon exposure to ATRA, all trans retinoic acid, you can see the GFP coming up. When you pack this into a virus and you infect the retinas of wild type or RD1 mice, these are PD6 beta mutant mice, knock-in mutant mice, you see that the RFP is more or less constant, but the number of cells showing GFP and the intensity of the GFP is increased. And this is a universal phenomenon. It doesn't happen only in mice, it happens also in rats. And we know that we have evidence, indirect evidence from dogs, from pigs, and even from monkeys. In any case, when the retina, the outer retina is dying, the inner retina gets flooded, gets inundated with retinoic acid, which activates its receptor. These models that I'm showing you here are global degeneration. They simulate mostly retinitis pigmentosa. We also wanted to know what happens in AMD. As you know perfectly well, mice and rats don't have maculas, but in a collaboration with Dr. Palanker from Stanford, we took rats, wild-type rats, regular rats, and we introduced a foreign object, basically an aluminum disc, in subretinally, which kills only the photoreceptors that are underlying that chip. And then we tried to assess whether retinoic acid signaling is increased more in the area underneath the chip as opposed to other areas of that retina. As you can see here, that in the degenerated area, there's a lot more GFP than in the control area, let's say around here, or even in the contralateral eye. And this happened for every single rat that we treated in the same way. So it doesn't matter which mutation kills the photoreceptor. It doesn't matter if the death of the photoreceptors is global or local, like in AMD. At the end of the day, the inner retina is going to sense a strong retinoic acid dependent signaling. Now, we want to understand what is the consequence of that, and we show here that RAR activation is necessary and sufficient to induce membrane hyperpermeability, that phenomenon that is associated with remodeling, with the changes in the inner retina. We can do that by chemical manipulation. If we take a wild-type retina and inject retinoic acid, we see an increase in dye loading, the same dye that is usually excluded from a wild-type retina. And the opposite, if we treat an RD1 retina with a small molecule inhibitor that blocks retinoic acid receptors specifically, you can see that upper loading is decreased. The same goes for a direct manipulation of the retinoic acid receptor. This is a form of the retinoic acid receptor, is the dominant positive. It is active whether the ligand is there or not. And this is a retinoic acid receptor dominant negative. This receptor will always be shut down whether there is ligand or not. You can see here that what happens is the exact mirror image, right? We have a lot of upper loading in the wild type, and we have almost eliminated all the upper loading in the RD1. These cells that you can see here labeled are pericytes of the vasculature. We don't know why they load upper, they always do. Doesn't matter, regardless of mutation or treatment. So we have demonstrated that it's necessary and sufficient for hyperpermeability, and here we move on to the most important part, which is the hyperactivity, because that is what is obscuring light responses. And both the chemical treatment with BMS, that small molecule inhibitor, and the gene therapy treatment with the dominant negative virus, both of them cut the spontaneous hyperactivity of RGCs by half. And this is important because then if you take an RD10 mouse, which is midway through degeneration and still has photoreceptors, still has some vision left, we can inject one eye with the vehicle, which in this case is PBS, and one eye with our inhibitor, and flash a light stimulus at the same intensity in both eyes, ex vivo, in an MEA setting. And you can see here that in the treated eye, we have reduced the background noise and allowed the signal to come out, to emerge from within the noise. This was done in 5 different mice in a blind setting. The person analyzing this didn't know which eye was injected with what. And in all 5 animals, we get the same exact response. BMS increases light responses. And this was just one flash of light at one intensity, but we can do the same, of course, at many, many different intensities of light. You can see that especially for not dim light, but the light that we use most of the time in our daily life, that difference is really strong, meaning that BMS is really enhancing the ability of the retina to reach all the way, for photoreceptors to reach all the way to the RGCs and that signal to go through. And this is all ex vivo in retinas that we dissect. And of course, we wanted to know what happens in vivo. And we did a few experiments with innate light responses, which tell us a lot about light perception, but they don't tell us anything about vision, about visual acuity, contrast sensitivity, and so on. And so the last part of my postdoc has been dedicated to try to build a system that we can measure visual acuity in a much better way. The mouse in this case is running on a wheel and facing a screen that can project any image that we want. And the mouse is trained to understand that only when it triggers the right image, there will be an available reward for the mouse in the reward area. You can see here how a young RD10 mouse runs on the wheel. The image will be triggered and the mouse goes right to the reward area and gets sugar water. But when the mouse ages and degeneration is complete, the mouse is blind and keeps running. The image is triggered. Here it comes, okay. And the mouse just keeps running because he doesn't see the image. And so what we can do here is basically we can train the mice in 3 or 4 nights to perform this task. You can see here that in one of the training nights, at the beginning of the night, the latencies for the response are all over the place, but through that night, the mouse learns, and the latencies to response, the timing that it takes for the mouse to leave the wheel and get the reward, gets shorter and shorter and shorter. Even if we take down the timeout to 10 seconds, the mouse is still performing perfectly because it takes only 2 seconds. But then when we take the same mouse at an older age, even with a 100% contrast image, we see, again, the latencies going all over the place. And that's how we differentiate between a mouse that is seeing and a mouse that is not. Then, of course, the test will be to change the contrast from 1 to 100% in discrete steps. The images are randomized. The mouse cannot know which image is coming up. Also, the wheel turns that the mouse needs to take in order to trigger the image, those are also randomized. And you can see here our metric of that is derived directly from the latencies. We call it response index, how that response index is affected before and after degeneration. So our goal, of course, now is to intervene and to use drugs. We are using several drugs. I cannot show you all that we are doing, but this is maybe right now the most interesting part. There is a drug called Disulfiram, also known as Antabuse. It's a drug that is given to alcoholics. I think not anymore, but maybe in some countries it's still used. This is an ALDH blocker. So ALDHs are acetaldehyde dehydrogenase family of enzymes. And in our retina, we have RALDH, the retinal form of ALDH, which converts retinaldehyde into retinoic acid, which activates the receptors for retinoic acid. And the advantage of this drug is that it's systemic. It crosses the blood-brain barrier, and we think it also crosses the blood-retina barrier. And we can provide the mouse with this drug in the food. What I showed you before, BMS, is a drug that we need to inject directly into the eye intravitreally, and that can cause damage to the retina. We can have some effects that we are less desirable, and also we never know exactly how much drug is in the retina after the injection, right? With this drug, we can be sure that we have a constant concentration, a constant feed of the mice with the drug. And here you can see that in a very small group of mice, only at one timing, 20 days after degeneration, we see that the mice that aren't treated with disulfiram, the reduction in contrast sensitivity is significant, even for high contrast images. But in the mice treated with disulfiram, there is no significant differences between before and 20 days later, and the responses are basically the same, meaning that we have preserved vision as much as we could. We, of course, are planning to increase this study to get more mice and to get also different types of tests that will test other things, not just contrast sensitivity, but other things like orientation tuning and so on and so forth. And with that, I will end here. I showed you that hyperactivation of RAR is necessary and sufficient to trigger pathophysiological remodeling in the inner retina of RD1 and RD10 mice, and also in rats with a rhodopsin mutation. And that includes both the hyperactivity and the hyperpermeability, especially in RGCs. Blockade of RAR activation or blockade of retinoic acid synthesis by disulfiram reduces hyperactivity, improves the signal-to-noise ratio, and the light responses that are initiated by the surviving photoreceptors can go through. Well, before they couldn't. The RAR signaling pathway is a promising target to counter degeneration-dependent remodeling, improve visual acuity, and boost vision in vivo. We are not saving the photoreceptors. The photoreceptors are dying. And for that, there are many other treatments out there and many other proposals out there. We are focusing on what happens to the inner retina as the photoreceptors die. This is a sharp change in direction from what is out there in the literature, and we think it might introduce a new approach to treating blindness in general. With that, I thank you very much. And of course, I have to thank my mentor and supervisor, Professor Richard Kramer, and everyone in the lab that takes a part in this great project. Bristol, Zach, Daniel, Mei Lee, who helped us with the viruses, of course, the students, and that's it.
Thank you. So we have time for one question. So there's a question on where the retinoic acid comes from, and if there's a difference in the number of RAR positive cells, that's green cells, between dorsal and ventral.
Oh, so those are two good questions that the answer are we don't know. So we suspect that the retinoic acid is coming either from the RPE or through the Mueller glial cells. And this is just an anatomic, based on anatomy, right? That's the answer to the first part. The differences in the ventral and dorsal and so on, by the time we test these mice, degeneration is global. In the assays, when we use the genetically encoded reporter, we used it always when the degeneration was global, besides the rats, of course, with the AMD. And we don't know right now, but there was someone in the lab working on that and trying to map the retinoic acid response across the retina from ventral to dorsal, from periphery to center, et cetera, et cetera.
Okay. And last question, what structural and functional differences are there in the inner retina supporting the fovea versus supporting the periphery?
So the differences in the inner retina, again, I can't answer periphery versus center, but I can say that on top of the hyperactivity and hyperpermeability, we know from the literature that there are changes, for example, in neurogenesis, the cells grow new neurites, they grow new dendrites. There are also cells that migrate and disrupt the layers of the inner retina. All of those phenomena, we haven't tested yet whether retinoic acid affects them, because we focused on the hyperactivity and hyperpermeability as very early on changes. The changes in the structure of the inner retina come much later in degeneration, many, many months later. These changes appear right away from the beginning of degeneration, and that's why we focus on them.
Okay, thank you so much. That ends the question and answer. Thank you. So we'll move on to our next speaker.